In today's session, we will cover molecular contacts and also have brief discussion of previous assignment. From the next session, we will be starting actual docking using some basic software. So till now you have a basic information of molecules. I will be gradually repeating everything again and again, so that if you are getting caught in somewhere or having any problem, you can share with me and we can discuss on that. So you will be getting knowledge gradually, not at once. In the last session, we visualized multiple molecules using different software. Today I will just give you brief about where you can find information or what you can find uh, in a in a software, or how you can interact with the software using its interface. So first I will open Chimera. I am not audible.
Yes, sir, you are audible.
Okay, someone has commented that I am not audible, so that's why I thought I am not audible. Okay, fine. So in the Chimera, you already know that this is menu bar here, and this is we use Microsoft Word. This is menu items and ribbons. So we can use these options directly, or similar options will be found in this menu bar. And whatever molecule you are opening, or whatever interaction you are having with this package, you will be having log over here. And if you are not able to find the log, it it will be stored somewhere under some menu in every package. Mostly you don't need to go for the log, but it is helpful sometimes. Next one is your models list. In other software, you will also be finding open molecule list somewhere, where you will be getting identification id number or something there, so that you can identify your molecule and interact with your molecule. So this is models panel. Similarly, in Avogadro, I'm just taking basic example of these two packages because these are open source, and other packages also work something similar. Here you will also get a menu bar, and this there are other menus which you can access directly. And molecular interaction options are present here for you. Display types or some of other options. Now we already visualized components of a molecule like in a single molecule there could be multiple other molecules. So how a molecular structure has other molecules, I will have brief about that. I will brief about that. I will again start with the where we started in the first session by going towards CSV. Random molecules. Like lastly, we open cyclooxygenase. Now, now, if you see the list, here you can see filters. This left panel. In this panel, we can get more information about how these molecules are prepared, or means about attributes of the molecules. Like if we are looking at some molecule, what attributes are present, you can have brief about that. So first of all, most of the structures are validated experimentally, and then second one is what experiments had been performed to get the structure, and next is what is the resolution. I will also explain this point as well. So next, release date means when those structures were published. Next is if there is any classification, if that is understood as enzyme, and then if that uh, protein structure or molecular structure is derived from some membrane. So these attributes are present here. Here I want to focus on this part, means how these structures are determined. There are multiple other methods as well. You can get all those methods from here, from this is advanced filter where you can search for uh, you can build your query, like if you want to find this molecule with some other things or other attributes, like what should be its resolution or any other property within that molecule, you can filter from here. So basically I want to know about experimental methods. So here what I will do, experimental methods, I will Google again. Whatever database you are exploring, there will be some statistics or other options which will help you understand how widely data is distributed in that database. Like if I am seeing RCSB PDB, here I can find out how many molecules are present and what are the experiments carried out to determine those structures. So this is the statistics, and here are multiple methods, like prominent methods are X-ray, NMR, and electron microscopy. So there are these many structures. So basically how these structures are obtained, like first you all know that sample is prepared, then those structures are bombarded with some radiation or chemical process to determine the position of electrons or atoms, and later they are constructed in three-dimensional space and submitted to RCSB PDB. So if we want to find structure of a single molecule like P53 protein, then if The method is performed using X-ray crystallography or NMR or any other method. Then, along with the particular molecule which is of our interest, there will be other molecules as well. Like protein is surrounded by water molecule, or it might be bounded to some other molecule. Or there could also be a case that while making preparation of the sample, there might be additional molecules to stabilize that sample. So apart from the molecule of our interest, there could be multiple other molecules as well. So in during visualization, last time we saw that there are ligands, or and along with protein, and there were other multiple elements as well. So basically, we also know that protein structure has amino acids, and amino acids are denoted by some names. So let us see what are the names of amino acids and how they can be uh, denoted. Protein, amino acids. Here is some list. So this is a standard list that we our amino acids are represented in protein. So in the structure, means whatever structure you are getting from RCSB PDB for protein, if you see any other symbol than this for the residue means if we are seeing the structure. So this is suppose there is some protein sequence, then it will get its primary sequence, one primary sequence. Then we will be getting amino acid one, amino acid two, and amino acid three, like that. If there are 1,000 amino acids or n number of amino acids, we can list them by like that, means in the sequence. So these are called residues, and these, and if they have sequence, they can be numbered, like amino acid one has number 1 2 3, like that they can be numbered. And later these residues have some denotation, like if we want to identify these amino acids, then we can give them three digit three character identification number, like for serine SER it should be present, for uh, glycine glycine should be present, like that all the amino acids would
Excuse
be.
me, sir.
Yes.
So there is some background noise.
Background noise.
Yeah, something like maybe sort of keyboard noise is uh, pinching when you are talking.
Screeching noise or something, or what?
Yes, sir, you are writing on the pad. That's why.
Oh, okay, well, okay, I will not use your pen then. It's a screeching sound or something. Okay, I will not use it. Okay, so the sequence of amino acids can be represented by like this. Let me show you last one file which we saw last. Now, my basic about this file lines. In a file, there are lines. I am just giving you overview of the file, means how you can understand what is the information inside the file. First, if you see all the content, all the text, all the content is divided, means has some break, a line break kind of thing. Means after every line break, on each line break, means after a line break, a paragraph starts, and so there are so many paragraphs here. If we talk about all these paragraphs, then each paragraph or each line represents a record. So this is one record, and this is one record. And what is this record about? That is denoted by first word, and after that there is some space, and then there is some characters which tell us the information about the record. So if we talk about this part, this whole line is a record, and this part this part is key, and this part is value. This is how PDB structure is determined. PDB information is determined by the software. So your software will be doing what your software is doing. It is going to check the file, read the file. It will read first line, grab the first word, and this is the key, means it will understand what is this information about. So remaining information will be parsed like that. So there are other records like compd and source, keywords, author, like that. There are so many records, means there is a whole lot of information there. Remarks are also there, but the major information stored here is the sequence or sequence of amino acids and their position. So I will go down. All these information means 6 rays, it's like that. This is also information about how three-dimensional structure is stabilized, means how other parts are interacting or how other parts are conformed in a three-dimensional structure. So first is here HET record. This is called het atoms, and there is hit name. So these records are related to means what other molecules are present. And later on I am looking for atom records. So these are the atoms, means what actually you visualize is atoms, means during visualization these atom records are parsed, and other information is just related to that protein structure. Like if we are having multiple atoms in a record or multiple residues in a record, if they are connected or disconnected, so all these information is are present in the package. So in this record, atom record, further we see that one there are multiple columns, means all those columns are separated by space. So first keyword tells that what is this record about. This is atom record, and there is one column that should be sequence of something, then another column it should be some denotation of something. I don't want to focus on that currently. Now this is residue record, means 3 character record. So if you see first column and the fourth column, then you can get the information that what residue is present, what residue is the means this atom is part of which residue. This is part of proline residue. So like that, these all are proline residues atoms. So the this whole information is about only proline residue. And next is its means sixth column is about their number, means like I told you that amino acid one must should have been given some number, and it should have some name. So this is proline name, and this is its sequence, and record name is atom and aniso type something. So basically you should be able to understand that if there is any atom record, there should be some it should be part of some residue, and then it should be it should have some sequence number. I will yes.
Are these coordinates for the structure?
Yeah, these are coordinates. I will explain all those things as well, but
Okay,
very
sir.
slowly over the time. Okay. So basically, just for today's point of view, I am just focusing on this part. Means if you see that a record is there, atomic record is there, then first there are columns, and in one column you will be getting what is this record about. That will be first column, and later on the information about that atom, and there will be one column where it will denote that this atom belongs to a particular residue and its name is that, and it should have some sequence that is 32 for now. I will explain all these things gradually over the time, because with hands on you will be understanding all these things easily. So like that, if I keep seeing going down, I will be seeing known residues like glycine, aspartate, glutamine, like that. All the residues we can see, and somewhere we will be seeing some unknown residues. Like if I see there is uh, on one hand we saw that there is atomic record, but here we are seeing that there is hit atom record. Hit atom record is heteroatom, means that is not the part of standard protein. So, or standard residues which are the part of protein. So here we are seeing that in the fourth column we are seeing HOH. It is some other molecule other than standard amino acids we saw here. Means it is not the part of this. So like that we can determine two things in a protein. Like first part is standard amino acids and non-standard amino acids, or uh, atoms which are not the part of protein. So all these types of atoms and residues we can find out in the protein, because since the structure of protein PDB file is that it should have some residue. So residue means that is the collection of atoms. So this is also given some residue name. Here it is for water molecule. So water molecule should have a minimum 3 to 4 atoms in its record. So if we see clearly, it is showing that oxygen is there, and it's for 12 number, means its hydrogen is currently missing. Means it should have hydrogen as well at the residue 12. So it is missing. Means this is the basic understanding. Now I will move on from here. So just what I want to make you understand, that if a protein is there in its primary structure, if a protein is there, then it will be having one degree structure, and in 1 degree there should be sequence of amino acids, and those amino acids have serial number, like we have roll number in our classes. After that serial number, 3 letter representation amino acid. So serial number is like 1 2 3 or something, and representation of amino acid is like 3 characters, some uh, X, X or like that. So 3 character representation of residues, and serial number will be some number, like some number, and then it will be starting from 1, or if there is something missing, it may start from other than 1. I will be opening one structure now for you. So in this structure, can anyone tell me how I can see the ribbon structure for this protein? Okay, I will just hide other options, hide and show the ribbon structure. Better I hide this one as well and show this. So if we see these means ends represent there is the presence of some atom. This is some atom, here one another atom, here another atom, and here another atom. So like that these are atoms at every nodes. We call it node and edges. Node means if there are connection of two lines, then this is the node. So these 3 points called nodes. Now we know that there were standard amino acids and non-standard standard residues and non-standard residues. So we can select standard residues and non-standard residues using some option like we did in the last session. From residue we will be selecting standard amino acids. So all the structures which are present here, which are selected are colored green, and we can hide them as well. So to hide those things we can click on hide and hide. So remaining parts which are not standard residues are now before you. If we hover on these these structures, we can get some information about them. Maybe I have uh, I have used control button and then clicked. So to get this information, control PRN plus click to select a atom or group of atoms. So like that you can select any atom. So that information will be visualized on left hand side, or it may be highlighted from here. Or you can also do one thing, that you can move to actions, label atoms. You need to go to actions, then go to labels, then go to atoms, and then select name or any other part you want to display. So what I will do is, now I will select one atom here using control and go to action, label, atom, name. So this atom is oxygen, means oxygen, and I will select other atom as well. Again oxygen. I show you that in the hit atom records there is some oxygen, but it is without presence of hydrogen atoms. So hydrogen is atom is absent from here. Okay, so like that you can separate standard residues with non-standard residues in a structure. Standard residues means which are known to the software, like it known, both of them are known, but known means part of protein or DNA. because for DNA or RNA you will be getting nucleic acid residues which should be having some other representation. I am seeing one white color ball here, so I will select it with control and go to select, sorry, action, atom and name. So it is zinc. We saw zinc is also present there. So like that you can get the overview of an molecule. And if I want to know about the position of this molecule, means in which what is the residue number of this atom. So basically I will go to action label, residue, and residue name and number. So currently I am seeing that it is overlapping. What I will do, I will go here to remove the labels. I will go to this models panel, open this, click on labels, and either remove it, or after selecting it I will click on hide or close. So like that I can remove that length labels. I will click on close, it is hidden. Now I will go to action label. I can label residue or atoms to know which name and to know its location. So I have clicked on this. So its residue name is zinc, and its serial number is 1342. So like that we can have the understanding about the presence of other components in a structure. Now we know that uh, in a biological system or in the world, two atoms interact, and if atoms are interacting they form molecule. First, if atom to atom interaction form molecule, uh, basically, and then molecule to molecule interaction form molecular contact or molecular interaction. Later on, if this is the part of biomolecule, then it forms biomolecular interaction. Like, and these biomolecular interaction further lead to biological process. Like we discussed last day, how does one antipyretic drug works. And these contacts could be like permanent. These contacts could be permanent or temporary. Like for the formation of a molecule, the contacts would be permanent, like ionic bonds, which are very strong and require high energy to be broken. So next one is that uh, for biomolecular contacts, there we know that there are weaker interactions, but they are in a large number, so they form a quite stable interaction, and form like they form tissues or other tissues, organs, and whole body or organ system, like that something. So, how does these interactions work? I am going to take one example of research paper, so that you can have understanding of how you are going to read some research paper. I will search for some like last day we talked about cyclooxygenase, and for fever we can use paracetamol, uh, yes, aspirin as well. So cyclooxygenase targeted by aspirin. I am going to look for a research paper. So this is an article, and if I am going to read the complete article, I am sharing this with you people in the group, so that you can read it as well. So now basically from here we read this simple paragraph. There are other so much information, and I just want to focus on a few things, means just to understand how we interpret the things. So here we are seeing, we can see that Uh, as- aspirin covalently modifies the enzyme. Means covalent interaction, which is still better than a few weaker interactions. So aspirin binds covalently, and in the last session we saw that some two drug molecules were binding and almost sitting within the three-dimensional space of the protein cyclooxygenase. So like that, aspirin might be also going to interact somewhere there. And similarly here, it is by transfer of its acyl group to serine 530. And what is this? These two information we have already seen that this is some serine residue and this is some sequence. So basically, with this line, we can know that this is the protein name, COX active site, and this is the uh, residue name and its serial number. So basically, we can understand that where this drug is directly going to interact with this protein. Means if this is protein, three-dimensional space, uh, amino acid will be having some number 1, 2, 3, 4. So if we go to 530th position in that protein, aspirin is going to interact with that residue. So we can also find these results in some of the docking papers and many other docking papers, which might be replicating these things. Now here acceleration in the block catalytic activity likely because increased. So basically, if it binds to serine 530, then it is not going to perform some of its activity, and further biological process is halted. And now here was just a single simple example. Next we can see if the same drug can target other molecules as well or not. Then in molecular targets of I think this will be having some brief information about this. So this is about drug. So other names of these drugs are here. Basically, I'm searching for its targets, mechanism of action. So it acts on all other molecules. Like it also interacts with cyclin A, PGG, H, protein kinase, tumor antigen P53, and NF kappa B inhibitor alpha. So these means drugs can interact with any type of molecule, like by it whether it is biomolecule like protein, or whether it is RNA or DNA structure. I am not currently seeing any DNA report here that where it binds, because we don't name DNA as uh, we provide gene a name, but DNA sequence is not provided a name. So we are, and also the research about interaction of gene and drug is like not that much prevalent. So we just see uh, protein and ligand interactions. So like that we can explore for the targets of our drug, or whatever drug we are searching for. Like if we want to target a particular protein which is responsible for disease A, and if we provide that drug to a person who is uh, having some other who is normal, and having that who is normal, then how that drug is going to affect that person, and how that drug is going to affect the person with that particular disease, where that particular protein is overexpressed or something. So by targeting the overexpressed protein, we can minimize the effect of a particular disease. Now we have seen that why what happens when molecules make contact with each other.
So,
Yes,
so we can repurpose drug with the help of this.
yes, there is like a repurpose. Existing drugs can be repurposed or modified, or we can search for brand new drug, brand new chemical which is not naturally occurring, or not has ever been known. So there are multiple approaches we can propose a drug, new drug, or repurpose a drug.
Yes.
Okay, so I will take one research paper, where what research paper I need to search for it. You people were having some research paper as an assignment, right? Can you provide me one link or something? Okay, you have provided already. I will see in the. I will take one example. Group binding nucleic acid. One of paper from the group member. Don't have access of it. Simply go to Sci-Hub. So this is one of the example paper, and here they have proposed that uh, some of the protein parts are interacting with nucleic acids. So what I will do is I will try to replicate this. This is very complex for now. Maybe I need to skip for now and search for some simple paper to find out here is this structure present. I need to look for the PDB ID. Is there some PDB ID? Maybe they have not provided me with PDB ID. Let me look for another some other paper. Only last random keyword is go modeling. There is some paper. Maybe it is also not. This is not also open source. Has anyone else shared their research paper? They are referring. Anyone else shared research paper in the group?
No, sir.
Complete article. I will directly look at PDB. So in this here they have performed some molecular document study and use this ID where they have some already existing ligand named and glucosamine, N-acetyl glucosamine. So I will open it in Chimera X by using open ID of the molecule. Uh, one one thing I have done wrongly is that I have not closed the previous structure. To close the existing structure, I am just going to models panel, selecting this particular structure which I have to close, and then I will go to this close option. Selected this, then close. So old structure is closed. Now I will go to biomolecule was hidden and this is the complete molecule. What I did, I moved to this graphics option and then view all. So they have used this particular structure and they are reporting them that there is nag or other residues are present. So I will click on nag and these glucosamines are selected now. And now if we see that how these are already interacting with the protein, we need to focus on single molecule. So let us see which is closer to the protein. Some random molecule I am selecting for now. I am clearing the selection by clicking this selection option which I have already told you all. So this will unselect everything. Now here I will select nag again. So what I'm going to do is in this graphics option I can restrict the view if I click this side view here, here, then I will get this panel, and this line, sorry, in this side view panel, I will get this line and this line. By moving these lines, I can restrict my view. I can restrict my view within this protein. If I move these two lines till here and here, then I can just see uh, this part, and other parts will be hidden or invisible or transparent. So what I will do is I will just move this gradually, so other parts are like not shown or hidden. Am I audible now? Hello.
Yes, sir.
Thank you.
Yes, sir,
Yes,
audible.
sir.
Yes.
Audible.
Thank you. Thank you. Thank you. I thought I am disconnected or something. So I will move the right line to restrict background view. So like that I can restrict the view and see my molecule clearly. Again I will click here, or I can also select this from select option, residues, and click here to select those structures. So like that I am seeing, I have focused on this particular single molecule. So I can see that here is some command again, and here again. For quick selection of some molecule. So I can directly use that those commands to select that, so that I don't have to navigate to menus again and again. This will make selection once again, and I will go to labeling, action, label, residue, name. So this has named residues, and I guess there are two residues. And then what I will do is I will now select nearby residue of protein by clicking control and click, and I will select go to again action and label, residue, and name. It is not showing any residue. Okay, it's also neg. I'm selecting some nearby residue of that protein, so that I can see display the name of that particular residue, so I can see that what other residues it is nearby. I will click again on name, so it is serine now. And here again some other helical structure is present. I will click on it and go to action, label, residues and name. So here name of the residue is VAL. I guess it is not, it may not be visible to you clearly. So what I will do is I will go to action, label, set label height. I will change it to 1. It is very large number. Maybe it's visible now. So basically here this nag means two energy residues are present, and this is written as VAL, and this is serine. So this serine residue is bit away from this residue, this ligand or non part which is not a part of protein, and this valine is nearby, means they are having some sort of contact. Basically in the information in the protein structure, you will be getting information about position of the atoms, but not how they are contacting. This additional information may be interpreted and added to the file, but this information is mostly not present. So in a structure, you will be mostly getting a how is a how is a protein constructed, means what are the coordinates or what are the locations of the atoms in a protein. So if this is some sort of structure, then it this point might be having some different position. Like if we take an example of our room, so in our room where you are currently sitting, there you might be having some distance from your front wall, from the floor, or uh, from the roof, and left wall. So like that, these that is called coordinates or some coordinate. So that helps us determine the position of atoms. So like if we talk about room, maybe I am sitting at some this position, so I am having some distance from this wall 1, wall 2, and from the roof there is some position of mine, so that is r1 something. So like that I am having some position, means if we talk about this distance and give it some number, like d1 d2 d3, then I am having some d1 d2 d3 position that is symmetrically presented as uh, coordinates. So if a protein structure is coordinates, the these coordinates just represent the atomic structures, sorry, atoms. So basically what information is present in the structure is that you will be having just atoms, means where one atom is present in the three-dimensional space, that information is with you for now. but how this information is interpreted, like if we see that we have named it as some residue, and we understand that if carbon interaction is there, carbon chain, and then it is later interacting with the nearby nitrogen is present, oxygen is present, or and some other group is present, which is representing some particular characteristics of a group, then we call it that particular residue or that particular uh, peptide bond. Like if their amino acid 1 is interacting with amino acid 2 in peptide chain, then the peptide bond will be having some characteristic feature that will be forming amide bond. So that characteristic is not present in the pdb file, means your software is showing you two atoms are connected, but actual information is just that, uh, just those points. The information about residues, their interactions, or how they are interacting is interpreted by you, or software does that for you for free, means without knowing, you don't know about that, but that does for you. So here these, like that one more residue is present here, and it is interacting with this somewhere here. So like that these are forming some sort of interactions. So interactions, if we talk about interactions, then that could be permanent or temporary. Permanent interaction, like amino acid sequence is there. So between amino acid there are stronger bonds, and that could be permanent. And temporary means which are continuously breaking and forming new bond. And if we talk about a biological process, there is some protein channel which is transferring something. So it doesn't just let some molecules slide in or slide out. What it does, molecule is coming to in its contact, it is forming some bond, it is moving forward, then it is old bonds are broken and new bonds are formed, and it is moving further. And this could be also, this could be one of type of interaction. And in another type, that is receptor protein which is interacting with the residue, means coming in its contact. When this particular ligand or particular biomolecule is coming in its contact, it is changing its shape, like that it's changing its shape, and it is changing its conformation, means it is either going to rotate, or it is either going to change its position. So like that it can do anything, means it can due to change in its shape, it might move from one part of the body to another part, and later on it may be moving to some physiological chemical gradient or something. So like that it can move to other place, and uh, where it might found suitable for it action. So by moving to other part, it will be changing its shape again, and these bonds which have been formed earlier will be broken. So like that molecules are performing their actions, and focus on we need to focus on that how two molecules are interacting. There are residues. Residues have atoms. Means residues are formed by atoms, right? So if I talk about R1 and R2, and there are atoms A1, A2, A3, A4, and here will be some atoms as well. Atom N1, N2, N3, N4. So maybe, and so to form bonds, atoms will be forming bond. So maybe this randomly two atoms are interacting with each other, and or some other type of interactions. So like that, only single interaction could be there, or there could be multiple interactions between two residues which I have shown in the PDB structure. So in the next session we will be dealing with I will also brief you about how you can visualize the interactions, means how these two group, means group 1 means this group or another group, how these are forming the bonds. So I will be, help you visualize that, means hydrogen bonds basically for now, and those, next thing is contacts. So these two basic things I will let you know that how you can determine or visualize these two types of interactions in a chimera X. So next and in the next class we are going to start with basics of basic molecular docking. For that what I will do is uh, we have a similar old assignment for you that we had in the last class. This is same as last class, and in the next session we will start with molecular docking. What I will do is I will give you ready-made molecules so that you can practice these visualization and interactions on real on molecule, which I will give you as example. Later on we will be taking talking about how these molecules are processed or prepared. So all those things will be discussed later, but I will give you prepared molecules so that you can just start with the docking and interaction. I will be repeating most of the things multiple times so that you don't have to you have means good practice about molecular docking and interaction thing. And I am expecting everyone to send me a research paper about docking they have chosen. So I will end this session today here, and if anyone is having any question, please let me know. Okay, let me just repeat one thing what you have learned today or what you have done today. First part is you can understand that there are residues, and what are residues? They are group of atoms. And second thing, that one residue interact with another residue. Interact means comes closer. What is happening, you need not you can explore on Google if you want, but for now, for this class you just need to know that in a contact or in an interaction of biomolecule, one residue is coming in closer to another residue, where along with that residue its atoms are also coming closer to that residue. That's it. And those residues have some identification, either we call it by name or its roll number or serial number or something. So like that you can identify a residue in a structure. Let me know if there is any question for from today's session. Am I disconnected? Sorry, I'm not able to read Google Doc.